So this is our lecture on biotechnology. This is chapter 20 in your textbook. Biotechnology has two main facets. The first is genetic engineering, which is where you process and manipulate genes and genomes. The engineering part is where you're actually doing the movement of the genes or taking genes out. We discussed earlier with the plasmids and the bacteria and how we make insulin and we insert our own type of genes. That's all forms of genetic engineering. Biotechnology is the process of manipulating organisms or their components for the purpose of making useful products. Biotech, the technology part, is the science behind it, and then we make products for sale or for use in the economy. Recombinant DNA, recombinant meaning recombination, that's the word you should think of, is DNA that has been artificially made using DNA from different sources. So for example, we talked about the plasmid in the bacteria. You take a human gene or a small series of genes and you insert that into an E. coli bacteria plasmid. And by that, we are able to change and manipulate the DNA, and the keyword there meaning recombinant or recombining. Gene cloning is the process by which scientists make multiple copies, either of an entire genome, of a specific chromosome, of a specific nucleotides of the DNA that they can then work with in the lab. So when everyone thinks of gene cloning, they usually think of Dolly the sheep and the whole organism that has been cloned. Gene cloning is just simply making exact replicas of genes. It could be a single gene, a small series of genes, or the entire genome as they did with the sheep Dolly. So gene cloning isn't necessarily and usually is actually just small portions of the genome. It's not usually the larger whole organisms. Some ways that they use genetic recombination, or the ways that they do these genetic engineering tasks, is restriction enzymes. We also call these restriction endonucleases. So remember, the ASES is always the enzyme part. They're used to cut the DNA at specific locations. We call these restriction sites. So that's how we're able to take out just a specific sequence of DNA instead of using the entire genome. Restriction fragments are the pieces that are then cut. So the fragment is cut with restriction enzymes. It's a piece of DNA that we're going to manipulate. And we say that one of these has at least one sticky end or a single-stranded end that we are able to attach. So think of it like almost like a Post-it note. There's the sticky part of the Post-it note, that small part that you're able to attach to the wall or a piece of paper. That's what a sticky end is. That's the part of the DNA that's going to attach to another DNA strand. DNA ligase joins the DNA fragments together. That's like the glue. It's the same ligase that joins, with the introns and exons and the splicing. That's the same ligase. And cloning vector is what carries the DNA sequence to be cloned. Usually it's a plasmid, that's circular bacterial DNA. The vector meaning just the avenue or the tool that is used for the cloning. Using a restriction enzyme, and DNA ligase to make recombinant DNA is shown in this picture. So you see at the top here is where the part that you're going to clone. So again, you're not cloning the entire DNA strand, you're cloning this specific sequence here. The restriction enzyme is used to cut the DNA at the restriction site. Notice it's not an even cut like here and here. You have the little tail ends that are called the sticky end. This is what is going to stick to the new DNA. You have the other parts that are kind of discarded as fragments. You bring in the new piece of DNA. So this is the outside DNA that you're bringing in. And then you go ahead and attach it, and notice the sticky ends. Notice it's a little off kilter. So this is where it's attached right here. And the ligase is what does all the gluing and the attachment. So the sticky end is what allows us to kind of fit in, almost like Lego pieces. They kind of fit in real nice so there's no interruption in the DNA. So this is how we would clone a gene. This is how the process is done. So you start with usually a bacteria, remember, because it has that plasmid that's easy to manipulate. You take a gene from what we call foreign DNA, or this is the gene of interest. This is the gene we're trying to study, and it's just a small segment of the whole DNA. We insert that into the plasmid as the process that we just looked at with the sticky ends. And then we take that new plasmid, we call it the recombinant plasmid, and we put that back into another bacterium cell, and then we allow the cell to function as normal. This is exactly how they make insulin. They take a bacteria cell, they take the code for insulin in the human cell, insert it into the plasmid, put that recombinant plasmid into a bacterial cell, and then the bacterial cell will actually start to make insulin that we then harvest and then we use as medication. Some other applications of gene cloning, which I know we've gone over before, any kind of biotech with the plants, gene and pest resistance, GMO plants, again, we talked about that, what they really are. It allows these plants to be resistant to insects and pests without having to spray them with chemicals. The bacteria that is used to clean up oil. We can make different types of human proteins or human molecules that we need, insulin, clotting factor, growth hormone. Those can all be made with bacteria much quicker than when we had to use real whole animals to make. Here's some other techniques of genetic engineering. PCR is a polymerase chain reaction. You can see here it's kind of going and cutting and splicing the DNA very much more quickly. You see up here, this is gel electrophoresis. This is how they do DNA samples. So if you ever look at like crime shows when they're looking at the DNA sample and they're trying to match the DNA, this is generally how they do it. It's kind of like chromatography. The pieces move down the gel and the heavier ones don't move as far, and then you can kind of match that up to get some DNA matching. And then the bottom one here is the protein, kind of using a synthetic protein and vectors and viruses to go ahead and correct a certain ailment in a human. So transformation is when the bacteria takes up the plasmid with gene of interest. That's when we actually, it's called transformation because the bacteria now has new DNA that wasn't original to that bacteria. The polymerase chain reaction, PCR, that we just looked at, amplifies pieces of DNA without the use of cells. Gel electrophoresis, again, was those DNA mapping that's used to separate DNA molecules using an electric current. And then DNA microarray assays study many genes at the same time. This is how we're doing the Human Genome Project and mapping the DNA strand. So here is the PCR, amplifying or copying a piece of DNA without the use of a cell. It's going to use the genomic DNA, and what you need to look at is there's only a small sequence that it does this for. So it's not going to copy the whole strand, it's just going to do a specific sequence. Gel electrophoresis, a little bit more information. It's going to use the polarity, so you have the little pieces here. This is where the DNA goes in the wells. This is actually a gel, a sturdy gel. You then attach it to a power source, and then the DNA will separate. And what you can do is match this up. So for example, you have an unknown assailant's DNA, and then you have someone that you think is the assailant. You can match up these DNA codes, and then they'll be able to tell you whether that was that person. And this is just a real life example, gel electrophoresis. They usually use some kind of chemical that makes them glow, but you can see here, this is the different fragments of the DNA as they had traveled through the gel. Microarray assays are used to study different genes. You use these, they're kind of very small well plates if you've ever used a well plate that have a lot of little holes that they use to fill. So we isolate the mRNA from the tissue sample, whatever you're trying to look at. You make something called cDNA using reverse transcription. Basically, you're labeling the DNA, remember, with the fluorescent, so it'll color differently. And then you apply the cDNA mixture to a microarray in each little well, and it'll hybridize with a new complementary strand, and then you can rinse and basically see where the fluorescent spot is expressed and where it's not, and you can kind of do this it's almost like targeting, like trying to figure out where certain traits are on a DNA range, kind of where it would look like. So this is the picture. So this is the DNA microarray that reveals expression of 2,400 human genes. So again, this is not that simple, but what we can do is each of these dots, for example, see all the red ones? Those are all the spots that would code for hair color, for example. And all the yellow ones would code for skin color, and the greenish ones would code for another, height. And then you can see some of these actually have multiple colors, which means they do multiple things. But this is what's helping us track the human genome and trying to figure out, okay, this is where this gene is coded. This is where this gene is coded. And they're kind of color coded so we can kind of match them up. Cloning organisms is something that has been done. It was a big kind of deal when they cloned Dolly the sheep. That is her name. This is where you do something called nuclear transplantation. You take the entire nucleus of a cell, all the DNA, and you transplant it into a body cell. So you're not taking a portion of the DNA this time. It's not a recombinant portion. It is the entire nucleus. You move from one cell to another. So you have the nucleus, and the memory cell now is inserted. It is then grown into an embryo, and then you get the full formed organism. It is genetically identical to the host nucleus, because what you're doing is you're skipping the meiosis, essentially. You're not making the gametes and having them recombine. You're essentially just taking the host cell or the parent cell, and you're just allowing that whole cell to form an organism. Some problems with reproductive cloning. The cloned embryos exhibited various defects. There still was some mutation that occurs. And the DNA of fully differentiated cells have epigenetic changes that again help in those mutations. So what they found is if you take a nucleus from a sheep that is 5 years old, well, then the chromosomes in that nucleus are 5 years old. So then if you regrow them from a new organism, an embryo, even though the baby is maybe 1 year, it's actually 6 years. So it's kind of aged in accordance with where the original cell DNA was. So that can cause issues where, you know, if you took it from a 20-year-old sheep, then at 20, the new baby is actually 40. So you're seeing a lot more mutations occurring with that, with the reproductive cloning. Stem cells are cells that can reproduce itself indefinitely and produce other specialized cells. Essentially, what a stem cell is right before the embryo starts to specialize. So remember, you have the zygote, which is totipotent, meaning it can be any cell. That zygote starts to replicate, it forms the embryo, which contains many different cell types, and then eventually your cells start to specialize. So what a stem cell is a cell before it has specialized. It's the point right before it starts to kind of differentiate itself into its different cells. So some applications of DNA, we can use them to diagnose diseases. If we really know where the location of some of these diseases are, it's easier for us to identify alleles and figure out where they are. Gene therapy, meaning we can possibly alter afflicted genes. So if we see that someone has the gene for breast cancer, we can take out that gene potentially and put in a new gene. And that has a lot of moral implications of whether we should be doing things like that. It helps us produce a wide array of pharmaceuticals that is currently in use now, as we talked about. All the different kind of hormones, growth factor hormone, insulin, those are all being made now with biotech. Forensic applications, so our DNA profiling getting better and better and better the more we understand the DNA and how to separate it. Environmental cleanup, so we talked about the bacteria that's helping us clean up oil spills and such. And then agricultural applications, GMOs, genetically modified organisms, specifically crops that are being made to be pesticide resistant. We're having them being made so that they replicate faster, so more food is being produced, and we're doing a lot of different things. So this is an example of gene therapy. So basically, if you take an RNA version of a normal allele and you let a retrovirus infect bone marrow cells that have been removed from a patient, the DNA can then make normal cells instead of making the mutated cells that exist in the bone marrow of the patient. Another example is what we call farm animal. This is a produced human protein secreted in milk for medical uses. Again, these are all just kind of farm technologies, animal technologies that we're using the Biotech for. DNA fingerprinting, specifically of children. You can go ahead and see the DNA of the child, see what traits they got. This is also kind of related to karyotyping. We talked about them doing that genetic counseling before you even have children. DNA fingerprinting again as well with the crime scene investigation, with forensics. You can go ahead and use the DNA analyzation. So the next thing is RFLPS. It's restriction fragment length polymorphism. This is where different restriction enzymes are used to cut the DNA. So each person has a different number of DNA fragments. And what you can do is take these DNA samples on the gel electrophoresis for disease diagnosis. You can also use this for DNA profiling. The old methods required almost a quarter-size sample of blood. This requires a very small fragment of DNA, so you're actually able to identify with DNA with much smaller amounts instead of having to do, almost like a whole blood sample in order to use DNA for profiling. So this is how RFLPS is used for diagnosis. You have normal hemoglobin and sickle cell hemoglobin. And what you would do is you'd use restriction sites in normal and sickle types, and then you put them in the electrophoresis, and you would be able to identify that this person has sickle cell allele instead of the normal allele. This can be used not only to diagnose the disease, but also to kind of, again, with the karyotyping, predict with genetic counseling of possibility of having offspring with this disease. STR is short tandem repeats. This is where you have non-coding DNA with sequences of 2 to 5 bases that are repeated. Each person has a different number of repeats at different locations. So it's another way that we can use DNA fingerprinting, but you only need 20 cells to get this type of match instead of, the whole blood, a good amount of blood that you used to need for DNA fingerprinting. Basically, you're actually mapping the non-coding spots instead of the actual code of the DNA, and every person has a unique non-coding, almost like actual fingerprints. That's kind of why they call it DNA fingerprints. You have your own certain type of ridges and stuff in your hand, and this is kind of the same thing but with DNA strands. This is an example of STR analysis, just mapping the loci, seeing if they match up, and then you would be able to tell if the DNA match or not. Again, just another look at it with 3 different people in the 3 different colors, and you can see whether or not they're matching up with the strand. GMOs, or genetically modified organisms, are any organism altered through recombinant DNA technology. Anytime you insert foreign DNA into a genome or combine DNA from different genomes. This is a huge hot button topic. The thing that people, I think, don't realize is GMOs have been used for years and years and years. Most of your cereal has GMO use plants in it, and now we're just kind of starting to figure out what they are. So then there's a grassroots movement of people who refuse to eat GMO food. Their argument is they're worried that the DNA that has been messed with is going to mutate their DNA, which is very, very unlikely. Actually, these genetically modified organisms are probably going to help with some of our food shortages. With famine in some places, it's really going to help us instead of hurt us. This is how you would gene splice. Again, you would take restriction enzymes and you'd cut it off, and this is just another example of a summary of the gene splicing in case you're not quite sure. This was in particular how they do it with a organism that is killed by caterpillars, and they're able to take plants that are then resistant to those caterpillars that are just the crops. Some other genetically modified food that you've probably eaten: corn, soy, cotton, papaya, rice, canola oil, potatoes, tomatoes, most dairy products, and peas. There are a ton of other ones out there, but they have been genetically modified for years and years and years, and for the most part we've been okay. So really, genetically modified food is a positive innovation. It is not a negative one.